Okay, thank you very much. Thank you, Matthias, for inviting me. It's really an honor to be here, and yeah, I'm enjoying this a lot. So thank you very much. It's really good to be after Max because he introduced many of the topics that I use, and I don't need to explain them again because now it's clear. And we are doing similar things, but here in Munich they are focusing more in the upstream analysis of the data. And I'm coming from Copenhagen, and we are more focusing on the downstream analysis of the data or interpretation of the clinical data, and we are using for that connective knowledge. Or what is the same is, or basically is just extracting knowledge from relationships. So this one, yeah. Going back to the workflow that Max presented, I will be focusing on these two steps. Basically the experimental design, because this is the door to anything that we do to get some or standardized data from the beginning, and then I will talk about the interpretation. But first of all, I would like to have a small introduction about how I met Matthias. So I met Matthias when I was still a kid, basically 3 years ago. But you can see that they were really tough years. And it's not much time to get to know Matthias, but I already know that he likes Lana Del Rey, which is some. He's crazy about tattoos because he's obsessed with mine. It's here, by the way, it's here, so. And then, and then you are all wrong. He is a real bioinformatician. It's not a mass spec guy. And then I have to say that what I liked the most about working in Copenhagen was that the objective that Matthias is defined at the beginning was that we should aim to change the world, to make a difference. And for me, that I'm an idealist, it was really motivating. And I know that some other people in the lab and myself, we saw ourselves in this drawing, Don Quixote. And I'm not saying that he's crazy. I'm saying that he was crazy enough to try to change the world, and also crazy enough to hire me. And now a less abstract objective was to move proteomics into the clinic. And as you could see from Philip's presentation, we already have some success cases, and we have many collaborations where we will aim to prove that proteomics can really help the clinic. But it's still, going back to the clinic with results is not so simple, and it's still challenging. And if you ask doctors, they always talk about these requirements, that the results need to be clear, interpretable, actionable, and quick. And they always mention these biochemical blood tests where you have different measurements and different ranges that define healthy. And if you are outside those ranges, that means that there is something wrong, and the doctors need to intervene in a way like with drug treatment or intervention with sports or food, for instance. But in proteomics and in other omics there are still challenges. And for me, the main one would be this one, that we have multidimensional data that then is a bottleneck in the interpretation or finding these actionable results. And normally we look at this multidimensional data as if they have different shapes, different colors, but then you can always bring into these data connections, how these data points are connected to each other, and these can help you identify patterns. Some of these connections could be protein interactions, pathways, functional associations, in general all the biomedical data that are out there. So we like focusing on connections, and if you look at this picture, it's basically a graph. And the good thing about graphs is that they allow you to define systems of interconnected components and provide also a biological framework that you can use to integrate different data types. in my opinion, they simplify the complexity by using some of the mathematical graph theory that those graphs have, and they also have these topological features that you can use to identify the role of specific components, how the information flows, and also you can test the robustness of the system. Also, graphs are visual representations of the data, and apparently human beings are very good at collecting data and finding hidden patterns using the vision compared to other senses. So that's another reason why to use it. Then we have all the data that is being generated, and it is known that we have exponential growth in the amount of data, experimental data that we can access right now. And it's not only proteomics, it's genomics, it's transcriptomics, epigenomics, and so on and so forth. And you also have many different layers of information. Could be medical records, images, physiology, or even the environment or exposome. So if you put all these together, that we need to interpret the data, that we have a lot of biomedical data available, we believe that graphs simplify and help interpret this data. If you put all this together with the experimental data we are generating in the lab, then you get this knowledge graph. And basically it's putting everything together. And so the aim of the clinical or the computational part of the clinical proteomics department in Copenhagen was set to build a database in the shape already of a graph that contains all the experimental data and all the biomedical data available out there. Well, it's not all, but the most useful one, or the databases that we normally use to interpret the data. The second step, or the second aim or goal, was to automate the analysis workflow with the current state-of-the-art methods, and then also extract all the power from this graph structure to help the users find information, but also focus on the relationships of in the data, integrate different data types, and also find hidden patterns, and also build new links or relationships using predictions. So we first needed to standardize everything, and for that we use different ontologies. In total, we are using 9 different ontologies that allows us to put the data, to harmonize all the data. And then we are integrating 21 different biomedical databases. And as you can see, we have pathways, we have protein information, we have tissue, associations, diseases, publications, metabolites, food, drugs, a little bit of everything you need to be able to interpret the data. This is the current status of the database. There are 16.7 million nodes and more than 113 million associations, 33 different node types, and 45 relationships. When we talk about the clinical knowledge graph, it is actually 2 modules. The first one would be the database up here, and that's the graph with all the experimental data and all the biomedical databases. But then we have a second part, which is all the code, Python code and R code that we are implementing to analyze the data, plot the data, and also query the data. And then we have developed 2 different ways of access this database or this analysis. One is through a web interface, and the other one is using the Jupyter notebooks. Oops, wrong. Yeah. So if going back to the workflow, the first part that I wanted to focus is the experimental design. And we use already the clinical knowledge graph during the experimental design, because you need to standardize all the data from the beginning, so you need to get the right identifiers. And in this video, you can see this is the web interface, and we are now creating a project. And in order to create a project, it's basically a form where you input different information, like the name of the project, acronym, who is responsible for that project, who is participating, what types of data you are collecting, and then what disease you are studying, which tissue you are studying, and so on. And once you have filled in this form with all the data, you create a project and you already get the right identifier for your project, your subjects, the samples associated to your subject, and then also all the associations to disease, tissue, and so on. So once this finishes, we create the project and we get this identifier. In this case, it's project 5. And if we go to the database, and this is an interface that the technology, the database technology provides, you can simply access the projects that are in the database. And as you can see, we have our test project right there. If I click twice, or in the video, I click twice, then you can see that you get all your subjects, all the relationships to which were the participants in the project, who is responsible, and also diseases, tissues, and so on. The second part would be the interpretation of the results. So if we have the experimental design, we run the samples, and then it goes through the through alpha-PEPT. Then we get output files from alpha-PEPT that we can format and put directly into the database, and then a process is triggered where we initiate all the analysis for the data. In this case, proteomics data. So here is how it looks currently, the interface. And this is basically the home page where you have some statistics about the database, number of nodes and types of nodes and so on. If you search for one of the projects, and in this case is one of the projects that Philip introduced, is the non-alcoholic fatty liver disease, you get these different tabs with information about the project. And one thing that you get here is, because all the data has been harmonized, the data for this specific project can be compared to other projects in the database. And in this case, it's returning two other projects that are very similar at the identification level, for instance. Then you go to the clinical tab, and here you find this tree that specifies the different analysis that we did on the clinical data. And these are parameters that we are using in the functions we are calling. So this is directly applicable to the code that we have generated. And here we have distributions of different variables, and you will see now also ANOVA and the correlation of the clinical variables. Then we do the same for the proteomics data, but we analyze the data specifically for the way we normally analyze proteomics. And so we look at different numbers of proteins, modifications, then we look at rankings, differential expression, and so on. And this tree can grow as you want, so you can add more and more analysis. Here's, for instance, the ranking, and because we have in the database curated markers, we can show directly in the ranking of the proteins if you found some of the known markers for the disease. These are volcano plots for the different conditions, and you also get correlation network, protein-protein correlation network, where you already find some clusters, protein-protein interactions, drugs, diseases, publications, and all the relationships you can get for those for the proteins that were identified, including also publications where these proteins were commented. Then in the multi-omics tab, if there are different types of data, we try to combine all of them. And in this case, we had clinical data and proteomics data, so you can run weight-gene co-expression network analysis that combines. It identifies co-express modules of proteins and then correlates to the clinical variables you had. If you had proteomics and RNA-seq, you could go for more network fusion algorithm, for instance. And let me just, that was, yeah, it went too far. Anyway, so we are planning to provide this as a container where you will get the database. You will also get the code that we have generated, and you will be able to just run this container, which will be a kind of a virtual machine that contains everything. One port to access the database, one URL that access the web interface, and one URL that access the Jupyter Hub where you have the notebooks. The good thing about the notebooks is that we will continue improving the analysis. We will find new ways of analyzing the data. We will find data sets for which the current analysis cannot work. So we will continue improving, and we will, once we make sure that this analysis or visualizations are good for the type of data that we want to analyze, then we just bring it back to the default analysis. So it's a continuous, it will be continuously improving. So as a summary, we try to build something that can harmonize all the data we are generating in the lab, that brings in all the knowledge that is already out there, and it also can help, based on the graph structure, can also help finding new patterns and make new predictions. We hope that in the future, and that this might be medium to long future, this could be part of the clinical setup. And we also believe that how we set up this platform can help analysis to be more replicable and reproducible, based on the fact that you can use the code that we have generated as you want, and then you can also use these Jupyter notebooks. And if we look at the picture at the beginning, so we went from data to knowledge to insight to interpretation of this data. And the hope is that by finding these patterns, we can already find some targets or biomarkers that can help in the clinic. And I would like to finish with this, that I hope in the next 60 years we focus on seeing the world as it should be instead of as it is. And with that, I would like to thank especially Anna-Rita and Anna-Laura that are working in this project, everyone in the group, and Matthias especially.